Every second, inside every living cell, thousands of chemical reactions are taking place. Sugars are being broken down, DNA is being copied, proteins are being assembled, toxins are being neutralized, and energy is being transferred from one molecule to another. But there is a problem: many of these reactions are naturally far too slow to support life on their own. Left to ordinary chemistry, some processes would take years, centuries, or even longer. Life solves this timing problem with enzymes. An enzyme is a biological catalyst, most often a protein, that speeds up a chemical reaction without being used up in the process. It does not magically create energy, and it does not change what the final chemical balance of the reaction will be. Instead, it helps the reaction happen fast enough to matter. In a living organism, that difference is everything. Without enzymes, metabolism would not simply be slower, it would effectively collapse. To understand enzymes, it helps to start with the basic language of a reaction. The molecules an enzyme acts on are called substrates. These substrates bind to the enzyme, undergo a chemical change, and are released as products. The enzyme itself is then ready to work again. This repeated cycle is why enzymes are so powerful. A single enzyme molecule can often process many substrate molecules over time. In cells, reactions rarely happen as isolated events. They are usually arranged into metabolic pathways, where the product of one enzyme-controlled step becomes the substrate for the next. This creates an organized sequence, almost like an assembly line. Each enzyme performs a specific task at a specific stage, helping the cell build, break down, or transform molecules with remarkable coordination. Metabolism is not random chemistry floating in liquid, it is controlled chemistry, and enzymes are the main tools of that control. The key to enzyme action is activation energy. Many chemical reactions are possible, but they require an initial energy push before they can proceed. Think of a boulder at the top of a hill, separated from motion by a small ridge. The boulder may eventually roll, but only after it gets over that ridge. In chemistry, that ridge is the activation energy barrier. Enzymes lower this barrier. They may hold molecules in the right orientation, strain certain bonds, stabilize temporary transition states, or create a local chemical environment that makes reaction easier. The result can be dramatic. Some enzymes speed reactions by factors of millions, or even more. A famous example is orotidine 5'-phosphate decarboxylase, an enzyme known for accelerating a reaction that would otherwise take an astonishing amount of time. With the enzyme, the reaction can occur in milliseconds. This is the central miracle of enzymes, not changing the destination, but making the journey biologically possible. Enzymes are also famous for specificity. Most enzymes do not act on just anything they encounter. Their activity depends on their three-dimensional structure, especially the region where substrates bind, often called the active site. The active site is shaped and chemically arranged to recognize certain molecules and reactions. Older explanations sometimes compare this to a lock and key, where the substrate fits neatly into the enzyme. A more realistic image is induced fit. The enzyme and substrate can subtly adjust as they interact, creating the best conditions for the reaction. This specificity is crucial because cells are crowded with thousands of different molecules. If enzymes acted randomly, cellular chemistry would become chaotic and dangerous. Instead, each enzyme helps maintain order by selecting the right substrate and promoting the right transformation. Specificity allows living systems to run many reactions at once without losing control. Most enzymes are proteins, which means they are built from chains of amino acids folded into precise shapes. That shape is not decorative, it is the enzyme's function. A tiny change in folding can affect whether a substrate can bind, whether a reaction can occur, or whether the enzyme remains stable. This is why temperature and pH matter so much. At low temperatures, molecules move more slowly, so enzyme activity may decrease. As temperature rises, reactions often speed up, but only up to a point. Too much heat can denature the enzyme, disrupting its structure and destroying its activity. pH works similarly. Each enzyme has conditions where its shape and chemical groups are best suited for catalysis. Outside that range, the enzyme may lose efficiency or stop functioning. Life depends not only on having enzymes, but on keeping them in the right environment. Enzyme activity is not fixed. Cells constantly regulate enzymes so reactions happen at the right time and at the right rate. Some molecules act as activators, increasing enzyme activity. Others act as inhibitors, reducing or blocking it. Inhibitors can work in different ways. Some compete with the normal substrate for the active site. Others bind elsewhere on the enzyme and change its shape, making the enzyme less effective. This regulation is essential for survival. A cell must be able to slow down production when enough of a substance has been made, speed up a pathway when energy is needed, or shut down reactions that would become harmful if left unchecked. Many drugs work by inhibiting specific enzymes in pathogens or human cells. Poisons can do the same thing destructively, blocking enzymes that are essential for nerve function, respiration, or metabolism. Control of enzymes is control of chemistry itself. The science of enzymes is called enzymology, and it has grown from basic observation into a major field of biology and medicine. Researchers study enzyme structure, reaction rates, regulation, mutations, and failure. This matters because enzyme problems are often disease problems. If an enzyme is missing, malformed, overactive, or inhibited, an entire metabolic pathway can be disrupted. Some inherited disorders arise because one enzyme in a pathway does not work properly, causing a substance to accumulate or a necessary product to become scarce. Enzymology also helps scientists design drugs, engineer industrial processes, and understand how life evolved. The deeper researchers look, the more enzymes appear not just as individual molecular machines, but as parts of larger networks. Their behavior depends on location, concentration, partners, signals, and cellular conditions. Enzymes are small, but their influence reaches across the entire living system. There are also important edge cases that expand the meaning of biological catalysis. Not all biological catalysts are ordinary proteins. Ribozymes, for example, are catalytic RNA molecules. They show that RNA can do more than carry genetic information, it can also participate directly in chemical reactions. This discovery was important because it suggested possible clues about early life, when RNA may have played both informational and catalytic roles. Another related field looks at pseudoenzymes, proteins that resemble enzymes but have lost catalytic activity. At first, these might sound like broken tools, but many pseudoenzymes still matter. They can act as regulators, scaffolds, or signal organizers, helping other molecules assemble or respond properly. Their altered amino acid sequences may remove direct catalysis while preserving structural roles. This reminds us that biology often repurposes old machinery. Even a molecule that no longer performs the classic enzyme job can remain useful in the larger system. More recently, scientists have also begun recognizing biomolecular condensates as another way cells can influence reactions. These are concentrated, droplet-like regions inside cells where certain molecules gather without being enclosed by a traditional membrane. By bringing reactants together, creating local gradients, or forming special interfaces, condensates can change how biochemical processes unfold. Their components may not be catalytic in the classic enzyme sense, yet the environment they create can still accelerate or guide. Reactions. This expands the older image of cell chemistry. Instead of imagining every reaction as one free-floating enzyme meeting one free-floating substrate, researchers now see that spatial organization matters. Where molecules are located, how concentrated they are, and what local chemical conditions surround them can shape reaction rates. Enzymes remain central, but they operate inside a much more organized cellular landscape. Enzymes are not confined to textbooks or invisible cell processes. They are also practical tools in daily life and industry. Biological washing powders use enzymes to break down stains that ordinary detergents may struggle with. Proteases attack protein stains, amylases break down starch-based residues, and lipases help break down fats. The reason this works is the same specificity that makes enzymes useful in cells. Each enzyme targets a certain kind of molecule. In food preparation, proteolytic enzymes such as papain are used in meat tenderizers. They hydrolyze proteins, breaking some of the tough structural components into smaller pieces and improving texture. In biotechnology and manufacturing, enzymes help produce antibiotics and other complex molecules under conditions that may be more efficient or selective than purely chemical methods. Humans use enzymes because nature has already solved a difficult engineering problem: fast, specific chemistry under relatively gentle conditions. What makes enzymes especially impressive is the balance they achieve. They are powerful but selective, fast but regulated, reusable but vulnerable to environmental conditions. They lower activation energy without being consumed, yet they can be switched on, slowed down, blocked, damaged, or repurposed. Their structure determines their action, but their action depends on context. This is why enzymes are often described as molecular machines, though they are softer and more dynamic than metal machines. They bend, shift, bind, release, and respond. A living cell depends on this flexibility. If every reaction happened too slowly, life would stop. If every possible reaction happened too freely, life would become chemical chaos. Enzymes create the middle path, controlled acceleration. They let cells move fast without losing precision, and that precision is one of the defining features of life. So an enzyme is more than a simple speed booster. It is a biological solution to one of life's deepest challenges: how to make the right reactions happen, in the right place, at the right time, and at the right speed. From metabolism to medicine, from RNA catalysts to pseudoenzymes, from cellular condensates to laundry detergent, the enzyme story reveals a basic principle of biology. Life is not just made of molecules, it is made of organized reactions. Enzymes are the hidden accelerators behind that organization. They turn slow chemistry into living chemistry. They allow cells to grow, repair, communicate, digest, defend, and reproduce. And although they are too small to see with the naked eye, their effects are everywhere. Every breath, every heartbeat, every thought, and every meal depends on chemical reactions made possible by enzymes. If you found this helpful, please like this video and subscribe for more explainers.